Hello, and welcome to the next session in our discussion about muscle tissue in Chapter 10. And what we're going to do today is we're going to talk about this structure, the sarcomere. And before we actually talk about that, let's review what we've been looking at so far. So in the last section, we broke down and understood the two major filaments that make up these bundles of proteins that we find inside the cell, these myofibrils, and we identified what is called a thick filament. And the thick filament is composed of about 300 or so of these molecules called myosin. Each myosin molecule is composed of two polypeptides that get wrapped around each other, and they produce this structure, which we call the myosin heads, and this long, thin portion, which is the tail. Notice that the tails are facing in towards the center, and the heads are out towards the edge. So this is my thick filament. The other filament that we talked about is the thin filament. This is composed of three different proteins: actin, which is this globular protein; tropomyosin, which is this long, thin protein; and then spaced along that long filament is the troponin. And you can see that spaced along tropomyosin. Troponin is the molecule that binds to calcium. When calcium binds to troponin, troponin changes its shape. That shape change then has the effect of causing tropomyosin to roll off the binding site on actin for the myosin heads. So what we're going to want to do if we want a muscle contraction is we want to connect that myosin head with one of these binding sites on actin. Right now we can't do that because they're covered up. But if we introduce calcium into the mix, then this molecule, tropomyosin, will expose that binding site, and so these myosin heads can now connect to that point, and we create what we call a cross bridge. Okay? So this protein is a contractile protein and a motor protein, because we mentioned that this head can come in two forms: the energized head and then the non-energized. And between them, we get this power stroke movement. So that's why we call that a motor protein, a contractile protein, because it's involved in actual contraction of the muscle cell. These two proteins, troponin and tropomyosin, there are regulatory proteins because they regulate that contraction. And then actin is a contractile protein because it forms the cross bridges with the myosin heads. So that's where we were with the last section. Now what we want to do is take these two filaments and put them together into a coherent unit, a functional unit, and that is called the sarcomere. So let's take a look at a typical sarcomere, and we'll do that first using the model. And so here is our muscle model, and we have these bundles of proteins. These are the myofibrils surrounded by the sarcoplasmic reticulum. And if we look at the longitudinal section, what we notice is that there is a pattern of dark and light and dark and light. As we know, that causes striations. When we look at these cells under the microscope, we get this pattern of light and dark, what we call striations. But this series of marks that you see right here is called a Z-disc. And then right here, that is also a Z-disc. Let me get two of these. So between my sticks is a sarcomere, and that's the functional unit of the myofibril. This is what's going to contract, or this is what's going to produce movement, what we call contraction. And each of these myofibrils are made up of many, many units of sarcomeres, one after the other, like a long train of cars, each car representing a sarcomere. So with that in mind, let's take a look at an image and relate what we've just looked at in the model to that image. So here we have a bundle that is a myofibril. We have one Z-disc we have another Z disc, and between those two Z discs is the sarcomere. So what we're going to do is we're going to take that section, and remember, the myofibrils are composed of many, many units of sarcomeres, one after the other, extending out, and we're going to expand that and look at the details. So as we look at the details, we can identify the thick filaments and then thin filaments. We've talked about those. The M line, which is right in the center of the sarcomere. Okay. And then at the edges, we have the Z disc. And then we have these other proteins, these structural proteins, which we talked about. This is titin, okay? And also myomycin, which makes up this protein here in the center. The entire thing, as I mentioned, is the sarcomere. And then we have zones within that sarcomere. We have H zone, we have A band and I band. Okay? And so what we need to do is define these zones and these bands, and then put the whole picture back together again to see how this unit is arranged. So let's begin. We've got the title of this section, which is sarcomere. And let's get some basic information down. So the basic functional unit of a myofibril is the sarcomere. It is the contractile unit. It's the unit that shortens. Okay. They are separated by the Z disc. Okay. And we have a pattern of overlapping thick and thin filaments. Okay. I'm going to write the definitions of each of these sounds, and then we're going to do a picture, an image, and identify those zones. So let's get the information down. First, we'll start off with the A band. A band is the entire length of the thick filament. The entire length of the thick filament. Let's come back to our image and let's just see what we're talking about when we talk about the entire length of the thick filament. So here, coming back to our image, the entire length of the thick filament represented by this term, the A band. So if I were to draw a line straight down here, it would connect up from this edge to this edge, the entire length, A band. Okay. The I band contains thin filaments but no thick filaments. The I band contains thin filaments but no thick filaments. Okay, so let's relate our statement to the actual image. And here is, so no, it contains thin filaments but no thick filaments. So here we have this area right here. We have thin filaments. Everything on this side of my finger is thin filaments. Everything on this side of my finger is thin filaments. And see, there's no overlap with the thick filaments. So that is my I band. And now I want you to notice that the I band crosses two sarcomeres. So let me draw that in a way that makes sense to you so we can get that visualized. So I'm going to draw two sarcomeres. And so this is my Z disc, my Z disc, and my Z disc. Okay. And then I'm going to come in here and I'm going to do some thick, 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 thick. Okay. And then I'm going to come in here and I'm going to do my thin. Thin, 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 thin. Okay. So now I want you to notice that our definition is I band contains thin filaments but no thick filaments. So if I draw a line here, draw a line there, that whole region, this whole region contains no thick filaments. So we have the I band. So, but in the center is my Z disc. So I've got a portion of one sarcomere, that's I band. Portion of another sarcomere, I band. Okay? So, thin but no thick filaments. Let's see. The H zone. The H zone contains thick filaments, but no thin filaments. So let's take a look and see what we mean by that. Thick, but no thin. So between that thumb and that thumb is just thick filaments, no thin filaments. So that's going to be my H zone. The M line is found in the center of the H zone. That is to say, the middle of each sarcomere. All right, let's go ahead and come back and look at the image. M line, center of the sarcomere, dead center of the sarcomere. So we've identified the sarcomere as between the two Z discs. The H zone consists of a region of only thick filaments. The A band is the entire length of thick filaments, so including that area where we have overlap with thin filaments. And the I band is only thin filaments, which can be found on the other, in the other sarcomere as well. Okay. All right. Let's take a couple of other images and take a look at them. And then we will do a drawing and lay out all of this information in the drawing as well. So here is an actual transmission electron microscope image of an actual sarcomere. So I've got my Z-disc. And now you can see light and dark pattern. That's what we've been talking about, the striations. In the center is the M line. The entire length of the thick filaments is the A band. Where we only have thick filaments and no thin filaments is the H zone. And then where only thin filaments are found is the I band. Okay. Okay. Another image. So here's my Z-disc. And now this is several sarcomeres now in position. And we also start to include other structures such as mitochondria, got the terminal cistern, the T-tubule. So we've got a lot going on in sarcoplasmic reticulum, but let's focus on what we're talking about now. So I have the H zone, which is just the area where there's only thick. I have the A band, which is the entire length of the thick filaments. Then I have the I band, which is only thin filaments. Okay, so dark, light, dark, light, dark, light. I have a whole series of sarcomeres. And what's going to happen, as we'll see, is this sarcomere, when we introduce calcium, will shorten. So instead of being this long, it will become that long. And the same is true with this one. Instead of that, it will all of them in the row that make up the myofibril will shorten together. Thus, the entire myofibril shortens, and we get a muscle contraction. Okay. So now what we want to do is we want to take this image, and so if you don't have this, you need to go get it. And I'll give you a second to do that. And we're going to go ahead and outline all of these things we just mentioned using this drawing. Okay. And then we've got one more image to deal with, and then that will be the end of this session. It'll be kind of a short session, and we'll move into the actual sliding filament mechanism. All right, so hopefully everybody's back now, and we're going to go ahead and identify all of the different parts we've just talked about. So let's see how we're going to do this. Let's start with identifying the sarcomere first. So I'm going to put a line here, put a line there, and I'm going to go here, and this is my sarcomere. Okay. And let's do pink. Okay. That's my sarcomere. Okay. Now I've got this, which is the entire length of the thick filaments. And that is going to be my A band. And let's do a nice bright color. a band. And then right in the center, here and here, I have thick filaments, but no thin filaments. And that's going to be my H zone. And let's get that color there. So that is my H zone. Okay. And then finally, I'm just going to draw half of the I band because I don't have two full complete sarcomeres. So we're only going to grab half, and that's going to be this section right here, and this section right here, and that section right there. And that's going to be my I band. And let's grab that in blue. And the definition of the I band is thin filaments only, no thick filaments. Okay. Let's go ahead and identify the components, the individual components. So that right there is going to be my Z disc. And that right there is going to be my Z disc. This is going to be my M line. I have thin filaments. And I have, and what color am I going to use now? Let's try orange. It's not very strong, is it? Let's see if this is any better. Okay. And that is my thick filament. Now, I'm going to do something, and this is to give us a better picture of how all of these are arranged. I'm going to take a section, a cross section. I'm going to make a cross section here. So I'm going to cut this, and I'm going to have two thick filaments. One, two. Two thick filaments. Okay. Now, I'm going to draw in the heads. So I'm going to have a head here. I'm going to have a head here. I have a head here. Because they're sticking out like this. Like petals on a flower. Now I got to draw the thin filaments. So first of all, let's use the same color to identify these structures. Okay. And now I'm going to do here, 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 here. That, or those, are my thin filaments. So thin filaments, thick filament. The point that I'm making is that these thin filaments are arranged in a circle, around the thick filaments, so that they're aligning with the position of the heads, because the heads are sticking out around the entire circumference of the thick filament, and each head is going to be aligned with the thin filament. Because what we're going to do is we're going to create those cross bridges, right? We want to connect that head to the actin in the thin filament to create that cross bridge. And then what's going to happen is all these heads are going to swivel in this direction when we get the power stroke. And because they're attached to the thin filament, they're going to drag the thin filament towards the M line. So this Z disc and this Z disc will collapse in towards the center as these heads are swiveling back and forth. And each time they swivel towards the center, they're attached to the thin filament and dragging it forward towards it, and we get this collapsing or contraction of the sarcomere. And each sarcomere in the line of sarcomeres that make up the myofibril is doing exactly the same thing, so the entire myofibril is collapsing or contracting, which, as we demonstrated earlier, is connected to the cell membrane, which causes the muscle to end up contracting as well. Okay. One last image to drive the point home. And it's this image right here. So we have a relaxed muscle, a partially contracted muscle, and a maximally contracted muscle. And what we want to do is pay attention to what's happening to each of these zones and bands. Okay? So keeping in mind that the H zone is only thick filament, the I band is only thin filament, and the A band is the entire length of the thick filament. And as we go from relaxed to partially contracted to fully contracted, what happens to each of those zones? Because when we start contracting, there's going to be a sliding of the thin filaments towards the center, and the Z disc will be moving towards the center. The I band and the H band will start disappearing. You see it getting smaller and smaller here, and finally it's no longer, neither of them are present, because now we have complete overlap of the thick and thin filaments. So there is no region where we only have thin filaments. There's no region where we only have thick filaments, because the entire thing has collapsed towards the center, and we get the contraction. So notice that this Z disc, that Z disc, is moving closer and closer. Its whole thing is being reduced in size. Okay? The only thing that remains the same is the A band, because that is the entire length of the thick filament, which does not change. It does not collapse. It remains the same throughout the entire process. What we're doing is sliding the thin filaments towards the center, and as we do that, the regions of overlap increase, and the regions where we don't have thin, or we don't have, we don't have thick, or we don't have thin filaments is reduced until, and finally, they entirely disappear. So in the next session, what we're going to talk about is the sliding filament mechanism, which is being demonstrated here. But what we want to know is the step-by-step process by which we can slide these filaments towards the center to get a muscle contraction. And that will be in our next session. So I will see you there.